Welcome to STEM Talk.
STEM Talk. STEM Talk. STEM Talk.
STEM Talk. STEM Talk.
Welcome to STEM Talk, where we introduce you to fascinating people who passionately inhabit the scientific and technical frontiers of our society.
Hi, I'm your host, Dawn Kernagis, and joining me to introduce today's podcast is the man behind the curtain, Dr. Ken Ford, IHMC's founder and Chief Executive Officer Emeritus, who is also the chairman of the Double Secret Selection Committee that selects all the guests who appear in STEM Talk.
Hello, Dawn. Great to be here.
So, Ken, today we have, would you like to explain today's guest?
Of course, Dawn. Today we have someone very familiar with STEM Talk listeners. That is Dr. Dawn Kernagis herself, a visiting research scientist here at IHMC and my wonderful co-host of STEM Talk for the past 9 years. Where did the years go? Dawn also has a new position as the Director of Scientific Research for DEEP, a UK startup that is pioneering a new era of ocean exploration, which is why the Double Secret Selection Committee invited Dawn for today's interview. For listeners unfamiliar with Dawn's background, she is a NASA-trained Nemo aquanaut, a fellow of the Explorers Club, and has been inducted into the Women's Divers Hall of Fame, and that just scratches the surface. Her research over the years has been focused on protecting the brain and nervous system of people working in extreme environments. Prior to joining IHMC, Dawn completed her PhD and postdoctoral training at Duke University, where she studied the genetics of decompression sickness and mechanisms of oxygen toxicity in divers. But before we get to today's interview, don't we have some housekeeping to take care of, Dawn?
As always, we do. First, we really appreciate all of you who have subscribed to STEM Talk, and we are especially appreciative of all the wonderful 5-star reviews. As always, the Double Secret Selection Committee has been continually and carefully reviewing iTunes and other podcast apps for the wittiest and most lavishly praise-filled reviews to read on STEM Talk. If you hear your review read on STEM Talk, just contact us at stemtalk@ihmc.us to claim your official STEM Talk t-shirt.
Today, our winning review comes from someone named Cheerful Flathead. It reads, I try to never miss an episode. I am a male of fairly advanced years and still learning cool things I can practice in my life. Thank you, STEM Talk. What a great podcast. I'm a retired man nearing 70. So many of your guests are fascinating and educational. But Abe Morgenthaler, episode 103, was special. I listened to the long podcast twice. I'm requesting a referral to an endocrinologist as soon as I return home. Thank you.
Well, thank you so much, Cheerful Flathead, and thanks to all of our other STEM Talk listeners who have helped STEM Talk become such a great success.
STEM Talk. STEM Talk.
STEM Talk. STEM Talk.
Dawn, welcome to the show.
Hi, Ken. Great to be here.
Very good to have you on the other side of the microphone. Dawn, you were our guest on episode 19, and you talked about your experience as an aquanaut aboard NEMO Mission 21. And for our listeners, NEMO stands for NASA Extreme Environment Mission Operations. And you, Dawn, have the distinction of being the only guest we've had on the show who we talked to from the ocean floor.
Yeah, that was a lot of fun. I still remember there's a point in that podcast where one of the crew members is outside of the undersea habitat that we were living in, and they jumped up in the window and scared the bejesus out of me, and I screamed, which I had requested that was taken out, but that was not taken out of the interview. It adds flavor and character to the podcast. But yeah, that was a very cool experience to be able to do that interview from the ocean floor while we were living there during the NEMO mission. I know we're going to talk about that a little bit more in this podcast as well.
Yes, indeed. It was very cool. As you well know, Dawn, we always ask our guests what they were like as a kid. I know in our recent conversation with Michael Schmidt, episodes 182 and 183, you and I talked about how we were both very curious children who actually could become quite annoying because we're always asking questions like, well, this is one I used to ask, why is the sky blue? Took quite a while to get an accurate answer on that one. Or, why is the grass green? Okay, got that one quicker. So other than being curious and annoying, what were you like as a kid?
Yeah, so I think the curiosity sometimes led to being annoying, and I found that fun, right? So when I would ask my dad those questions, well, why? Eventually he'd be like, okay, this is enough. We're done with this game. So But yeah, just super curious. I read tons of books. I loved visiting science museums. I would just geek out at all the different displays. Was fascinated by cats and mummies, archaeology. And this is when I was in elementary school, dinosaurs, and then I really got into rocks. Actually, for a little while, when I was probably like 6, 7, 8 years old, we actually had my dad purchased a rock polisher, a rock tumbler.
Oh, yeah. Lapidary.
Yeah, so we ended up, I would just collect rocks and we would polish them. I still have some of those to this day, but it was cool that my parents really supported me in those endeavors. I think I never asked them, but I'm wondering if they thought it was a little weird, but, but I was getting into things that were relatively healthy and just found everything fascinating. So lots of questions and just, digging into anything that I could get an answer to.
Yes, I too went through the rock stage and polishing rocks and all of that. And I hadn't thought about it for decades. I mean, many decades.
Mm-hmm.
And I'm spending a lot of time in Wyoming these days. And it turns out Wyoming is heaven for rock hounds. And they actually have shops in these small towns that have nothing in them but rocks of all different kinds. And some of them are raw and some of them have been polished. And I can imagine as a child, if one of us had seen a store like that, it would have been shocking. I had never seen stores full of nothing but rocks.
Yeah, that would have been absolutely fascinating. I can tell you, I spent a lot of time at those science museum gift shops where they would sell those rocks.
Yeah, the little rocks.
Yeah, asking for my parents to buy some for my collection. So yeah, that would have been very, very cool. And Ken, we'll have to compare, what our rock collections were at some point too.
You probably beat mine, but I had a lot from Cuba and Eleuthera
cool.
and places like that where I lived as a child. Now, I know that in your youth, you spent a lot of time in North Carolina, but you also lived in some other places as a child, particularly a young child. Could you tell us about that?
Yeah, absolutely. I was born actually in Fort Collins, Colorado. So my parents actually met out in Colorado, and I lived there until I was pretty young, so until I was about 1.5. And then we went to Iowa. I actually had a medical emergency that took us to Iowa. And so I was at Iowa Children's Hospital, and eventually my parents transitioned there to Waterloo, Iowa. And my dad actually worked for John Deere for quite some time as an electrician. And then we ended up leaving Iowa and moved to Raleigh, North Carolina, where I've lived most of my life on and off again ever since. So that was when I was about 8 years old. We moved to Raleigh.
So, as I mentioned in the introduction, you were inducted into the Women's Divers Hall of Fame. So when did you first become interested in diving?
Yeah, so essentially when we moved to North Carolina, so I was 8 years old, we went down to the coast of North Carolina. And actually, I'd been to the ocean once before. That was the Pacific Ocean when I went out to visit my uncle. And I have pictures of me standing there with my dad, looking out over the ocean. I love that picture. Fast forward to when I was 8, and I went down there, was swimming in the water as I always did, and just really immersing myself, and couldn't get me out of the water at the beach. But it was really the experience of going to the Fort Fisher Aquarium that's on the North Carolina coast and seeing all the fish and just seeing all the different species that live subsea, and that curious Dawn that was fascinated by rocks and cats and mummies and everything else said, hey, this is amazing. I want to become a marine biologist. And my dad had actually taken an oceanography class, had been taking that at North Carolina State University. And so he had some oceanography books. So I started pouring through them like I understood what they meant when I was 8 years old. I wasn't a child genius or anything. So I think I just like to look at the pictures and the graphs. But I was like, this is what I want to do. So that's when I first became fascinated with scuba diving, realizing that if I want to be a marine biologist, I should learn how to scuba dive. And so started just reading all of the diving magazines. I would actually write essays about what I thought it would be like to scuba dive when I was in middle school. Yeah, I was just ready to go underwater. My dad finally took us to the Florida Keys for the first time when I was in middle school. He took my best friend and my sister and I. That was my first snorkeling trip. And we were driving down the Keys and I could see the coral reefs as we were driving over one of the bridges. And I yelled to my dad, stop the car. And he thought something was wrong. I was like, no, I just want to look. And he's like, okay, don't do that again, especially if we're driving on a bridge. But yeah, so that just like sealed the deal for me. I was like, yep, I'm just chomping at the bit to learn how to scuba dive. That's where the passion first started.
Yeah, that does sound pretty great. Do you recall how old you were when you started actually scuba diving?
Yeah, so I was 15 years old when I first started scuba diving because you could get certified a little bit younger, I think at the age of 12, but you'd have to go through with a parent. And my parents are both just super busy working multiple jobs at the time. And so I waited till I was 15 when I could take the class by myself.
Yeah.
And I pretty much didn't leave the local dive shop. It was called Bill's Scuba. I pretty much didn't leave there after the fact. So, yeah.
That 15 sounds actually to me like a better age to take it up
Yeah.
because, first of all, there's nothing worse than having to do something insanely cool with your parents. But I think 15 is, in many ways a better age.
Yeah, and I understood some of the science and math behind it too, just some of the basic physics you have to learn. So I'd been learning that already, and of course had been reading every book I could get my hands on up to that point. So I think if I had done it younger, I may not have understood those things as comprehensively as I did when I was 15.
I'm sure that's true.
Yeah.
So after high school, I know that it was your plan to attend the University of North Carolina, but you ended up at North Carolina State. How did that happen? And, these stories are always really interesting.
Yeah. So I was accepted to University of North Carolina and planning to go. I was going to roommate with my best friend. We met the day after I moved to North Carolina when I was 8. So, this is the next step for both of us. And I was doing a lot of cave diving at the time. So diving underwater caves, especially in Florida, and had gotten involved in some cave exploration projects. So there's actually one that was in the Ozarks that I was participating in the summer before I would have started at University of North Carolina, and eventually got invited to participate in a cave diving project in Mexico. And I was like, I can't turn this down. And so I went to UNC and I said, hey, if I've got this project in hand, I would start in the spring. Are you guys okay with that? And I thought that would be the case. They would say, yeah, absolutely. Start in January of, that would have been 1996. And essentially they said, no, you have to start in the fall. And they wanted, their classes to start at the same time. I get that. But essentially I was like, well, I'm not going to wait a whole year to go to college. So got back from Mexico. And in the meantime, I had applied to North Carolina State University, which actually ended up being a better fit because my background is definitely more science, a little bit kind of an engineering background. So I think even culturally, NC State was a really good fit for me at the time. And so I was really happy with that decision to go to NC State and become a Wolfpack fan instead of a Tar Heel fan.
Yeah, they're both quite amusing names.
Yeah.
Wolfpack and even better, a Tar Heel, right?
A Tar Heel, yeah.
At first, you wanted to major in physics, I understand, then marine biology, and then biochemistry. And I've heard you say that there are probably another 10 areas of study that you would like to do if you had 10 lifetimes in which to do it. Can you talk about this wandering path? I suspect many of our young listeners in particular who are in college or graduate school or thinking about college will find your journey quite interesting.
Yeah, and I'll say that 10 areas of research has probably expanded even more so since going on STEM Talk and working with you on STEM Talk, with all of our guests who do just such fascinating work and are leaders in their fields. I'm like, oh, that sounds like such an amazing area to do research in. But anyway, I love what I do, so I'm not taking any of it back. But yeah, I first started off, I think physics is because, I was pretty heavily involved in physics from the diving perspective. So dove all the way from late middle school all the way through high school. So I was like, oh yeah, physics makes sense. And then I took a class because I thought, again, everything was fascinating. And just like the little kid in Dawn, I was like, oh, I'm going to take a marine biology class. And it was actually a class focused on evolutionary biology with a kind of a marine biology perspective. And the professor, John Godwin, just was an incredible professor. I was like, I want to do this. So I changed my major. That was pretty early on. That was like within the first year of undergrad. So I kept on clicking along and then ended up just, again, being fascinated by a lot of different topics, kind of ended up in this kind of pre-med concept because all these people were like, oh, you really should go into medicine. That's what you should do. And I could see diving medicine as maybe an area of interest for me. But again, was interested in everything and actually worked on some marine biology projects as an undergrad as well. But yeah, I landed on biochemistry because it felt like it was a good catch-all, from physics to chemistry to biology.
It is.
Yeah. And it was just a really good kind of, broad spectrum degree. I also had, again, a really good professor or two that I was able to work with, including Dennis Brown, who was the chair of the biochemistry department at the time. I actually ended up doing some virology research with him in the biochemistry department. So yeah, I would say follow your passion, because at the end of the day, I think you've probably picked up on this, and this is to the listeners. A lot of our guests don't end up doing exactly what they majored in. I think that first major is kind of setting the stage, maybe with engineering a little bit less so, but you're kind of setting the stage for the future work that you do in your life. So follow your passion is probably my biggest piece of advice.
Exactly. And make sure that passion is something you're actually good at.
Yeah, that too. That's important.
So, following up on that, you mentioned diving all the way through high school, and as an undergraduate, you were doing lots of cave diving. That you mentioned the trip to Mexico as well as other places. You and your friends were doing these deep dives, 6 and 7 hours at a time, and I think that's when you started to become really focused, you might say obsessed, with ways to protect yourself and your friends who were diving in these extreme underwater environments. And at that time, there wasn't really that much research on these topics, was there? And could you tell us about these long dives and why it was necessary to be thinking about this?
Yeah, no, it's a great question. So I eventually started, so I talked about cave diving through high school and eventually became involved with a project called the Woodville Karst Plane Project. So I was a diver with them, but also I was operations manager for them for quite some time. I actually worked with them for over a decade. And they were setting world records in underwater deep cave exploration at the time. They were really experimental in their, the way that they are conducting their dives, the equipment that they're using. There's a lot of equipment now in the scuba diving industry that evolved from changes that were made specific to that type of diving and even specific to our team at that time. So it was really cool to be in the middle of this kind of revolution for deep technical diving, especially for cave diving. There's even team training, other types of methodology that's changed since the WKPP first started. And as they're conducting these types of dives, it was necessary, right? We're doing these really, really deep dives with people from all over the world that were team members. And so we needed to have kind of a comprehensive way to keep people safe, be interchangeable when they're working across different teams. So all of that was kind of addressed, right? So we addressed that from the training standardization with the novel techniques and equipment that we were developing. But the biggest gap for us was on the physiology side of things. And again, it was really pushing the limits to the point where Frank Butler, who we've also interviewed on STEM Talk, came over. He was at the Navy Experimental Diving at the time. He actually came over to the site to see, like, what are you guys doing? How are you guys, doing these dives out in the field? It was very different from the way that the Navy would conduct dives like this. I mean, they were also doing deep, long dives, but the way we were doing it was very different. And we were working with scientists and folks that were really helping guide us on it. But again, it was still very experimental. And so at the time, jokingly, like, we would have papers, we would dig into some of the literature. And it's like, eat more blueberries for antioxidants to protect the divers and things like that. I was like, well, you can only eat so many blueberries, especially before you do a long, deep dive, by the way.
Yeah.
You probably don't want your stomach filled with blueberries. But anyway, so it just really kind of started to open up questions for me of like, what we saw is some individuals on our team seemed to be more or less susceptible to decompression sickness. We saw that some people got really cold when others didn't, even though they're wearing the same equipment. So just go down the line. Like, there's these differences within individuals that was really fascinating to me. And then bigger picture is like, if we're going to keep pushing the limits on this, it'd be really nice to know, like, are there ways that we can make our divers safer while they're doing these really long dives? And again, we're talking, for anybody who's scuba dives on the podcast or has familiarity with it, 300 feet average depth for 6 to 7 hours. So you're looking at almost a 24-hour decompression coming back up to the surface to minimize the risk of decompression sickness or the bends. And so this is a lot of time in the water. These are really high stress dives. And so, so that's what led into my interest in kind of digging into how we could better protect our divers.
Could you talk briefly about how these long decompressions were actually handled and staged?
Yeah, that's a great question. So we had support teams that were around the clock. So I said that 24-hour decompression. We had people that would actually stage backup gas and backup scooters and all the things that our dive teams were using into the cave. Subsequent weekends, we'd be pulling all that stuff back out of the cave. But you'd have these caches for the divers to use and also for safety, like as they came through the cave. And then for the decompression itself, they were diving what's called trimix. So essentially, it's a helium, nitrogen, oxygen mix. That was newer at the time. So before that, we were adding helium to the mix, but we really got to using it more and more throughout our dives because we learned, how to use it safely. So the reason you add helium to the mix is to negate the narcotic effect of nitrogen, especially at depth. We're doing these really, really deep dives. But the thing with helium is that when you're decompressing, it has different characteristics than nitrogen does. So we knew a lot about the nitrogen characteristics, but a little bit less about the helium characteristics. So there's still a lot that we were all as a community learning about how to do these trimix dives safely and decompress from those. So as the divers came out from a deep dive in the cave, they would slowly but surely come up and spend, minutes to hours at different depths, kind of making their way up by 10 feet at a time. And over time, the gas mixes would change where they would have more nitrogen, less helium. So it would help with that pressure differential. It would help off-gas that nitrogen from the diver's body. And then as you got even shallower, so like around 30 feet or so, we would start to switch to a higher oxygen concentration, actually to pure oxygen. But once you get to that point when you're breathing pure oxygen at depth, you have a risk of oxygen toxicity, which I know we've talked about with Dom D'Agostino in previous podcasts. And so this is where you can, one, there's a pulmonary oxygen toxicity that just irritates the lungs. But the more serious one is central nervous system oxygen toxicity, where you can develop seizures underwater, which is very deadly. And so our divers would go into a makeshift habitat. They were overturned troughs or bins where we had built a floor into those bins. And so the divers could go and sit up in the bin and breathe their pure oxygen and be in a dry environment. So if they did seize, they would be in a safe environment to do so. And so they spent a lot of time in those dry environments, but they were also able to get up in there and eat and drink and, communicate back with our team to the surface. We actually had comms relays back and forth to the surface at that point. And then they spent a lot of time there before they finally came up to the surface. So it was a very long process that involved a lot of support as well. So divers just making sure that all the equipment was there, that we were pulling the used equipment out, that the divers were safe, and if there was an emergency, that they could handle those emergencies at depth. So again, these big dive weekends, just for perspective, you would have 40, 50 people on site rotating through for different types of diving tasks over a single weekend. And this was all volunteer, which was actually pretty cool that you had these people coming in globally to participate in these world record breaking dives.
Yeah, it is for sure cool. And so based on these experiences, and I'm sure you were also scouring the literature for the latest relevant research, you came up with what you thought was a great research idea. And you scheduled an appointment with Dr. Richard Moon, who is one of the leading hyperbaric medicine specialists anywhere. Richard was actually our guest on STEM Talk episode 26. Do you want to talk a little bit about what happened and how the discussion with Richard went and your idea?
Yeah, absolutely. So, and it's funny, I had known Dr. Moon for quite some time because living in North Carolina, he was at Duke University. They would run studies both with Duke and also Divers Alert Network, otherwise known as DAN, doing dive medicine studies. And so they would always be looking for volunteers, participants for those studies. And I was often a participant in those studies, just out of my own personal interest and wanting to contribute back to the community. So I'd known Richard through that process for quite some time. I was actually really nervous. I had written out, I remember I just had pages and pages of notes. I was actually interested in looking at different pathways, so immune pathways, complement activation, inflammatory pathways. So I was just digging through all the literature up to date and actually doing this from a library, which I don't think anybody does anymore. So I was actually pulling papers in the library and making photocopies and sitting down and immersing myself in the literature and trying. I actually remember there's a diagram of like showing how all this stuff tied together and like maybe this is how the body's responding to bubbles that are in the blood and tissues. And maybe this is setting off these pathways. And I had all these different little kind of mini hypotheses that I took to Richard. So I set up a meeting with him and I was so nervous. It's so silly because I had known him for quite some time, but I was getting ready to pitch a concept to somebody I just had so much respect for, right? And Richard's the kindest soul. There's no way he's going to be mean about it, even if it was like the dumbest idea. So he's just a really good human. But I went in there, I just remember shaking. I was like almost ready to throw up. I was so nervous when I went to present to him. I don't know if he even knows that, but I went in and pitched it and he's like, yeah, I like this, but we're going to need funding to do this study. I was like, oh, okay. So how do you get that? And I had written like a mini grant before to Sigma Xi when I was an undergrad for some marine biology research, but I didn't really wrap my head around like what a true grant proposal process looked like. And so he said, well, you're going to have to write a grant proposal. There's a bunch of examples that I have. So I'm going to share one of my successful grant proposals with you so you can see what that looks like. So first off, you have to write a pre-proposal. So this is all new to me, right? Just coming out of undergrad and having worked in the diving community for a little bit at that point. So then we were talking about like when I could submit that grant proposal. And actually one of my cave diving teammates on the WKPP also worked for the Navy Experimental Dive Unit and was a researcher there. And so I had reached out to him just for some feedback. And he said, yeah, there's actually a proposal deadline coming up. It's in a month. So I wrote my first pre-proposal, did it sitting on the beach while I was working on another job that I had at the time. And we submitted it. It got invited for a full proposal. And once I knew it was invited for a full proposal, I actually went out to talk with the Navy. And I said, if this gets funded, would you all be interested in funding me as a graduate student? and this becoming my graduate research. And they're like, yeah, that'd be fantastic. Because at the time we knew that there was a big gap that was going to be growing in the dive medicine community, especially from the research perspective. And they knew that they wanted young talent coming into the field. And I say young, it's all relative. I was in my 30s at the time. Elizabeth Montcalm-Smith and Matt Swergas were the first two program managers that I worked with at the Office of Naval Research, the Undersea Medicine program. And I'm thankful that they actually believed in me and said, yeah, let's turn this into a pre-doctoral project, pre-doctoral funding, essentially, that became at least a part of my project for my PhD work. There's a whole other slew of things that happened after that, and I think we're going to talk about that in a second. But I'm super grateful that Elizabeth and Matt saw the vision and believed in me to fund me as a pre-doctoral, not even a candidate at that time, right? I wasn't even in grad school yet.
Right.
So, and then Richard Moon being part of that definitely had something to do with it. But Richard was always fantastic. Like, hey, this was your idea. You saw this one through. So again, just really thankful that they had that vision. And it's been really cool to see because since then, through that mechanism, they're like, okay, Dawn's going to be our test case. If it's successful, then we'll fund other folks. And they have funded other predoctoral students through that mechanism since then.
You must have been one of the very first, I would imagine.
Yes, I was the very first for undersea medicine for sure. I'm not sure about O&R totally, but for undersea medicine, it kind of set that pathway up.
I can imagine. You mentioned going to the library, and lest our youthful listeners feel sorry for the aged among us, when you had to actually go to a library, it was not a bad experience. And when you found that special paper, the one that really was helpful, it's like you found a treasure, in this huge place full of a couple million books and who knows how many journal articles.
Oh, yeah.
And you find just the one you needed. It had a different feeling than doing a really clever search online. Maybe I'm just showing my age and preference for paper, but it was sort of a different feeling. Did you experience that, Dawn, or is that just my delusion?
Yeah, absolutely. No, and I'm the same way. I'm very paper oriented, so I might be biased as well. But it's not just that. It was being surrounded by all of that knowledge, right, and all of that history. And so there's just something about being in this super focused environment where you're there. Like that's the reason you're there, is to find it, soak it up, integrate it into whatever it is that you're thinking about or what you're trying to plan. And I just would spend hours of my free time going over to the Duke University Library. And this is before I was a grad student. So I was just going in there just as a visitor and would just have books stacked up. And yeah, every once in a while you'd find that paper, which would then lead you down a whole other rabbit hole. But I just remember having those papers in your hand and you just carry it with you everywhere. And just being in that environment, I miss it. I miss having that. And I actually, even to this day, sometimes will go to one of the university libraries here in North Carolina if I really want to focus, or if I am looking for a little bit of inspiration, if I'm writing or something. This is just such a great environment to do so.
That's exactly right. And I still recommend that people actually physically go at times to a scholarly library, and it shifts a little bit your thinking. I think it's helpful. You discussed the path you took, and you discussed the research and your beginnings with the Office of Naval Research. But as I recall, your thesis ended up being more about cancer than about diving-related research. How did that come to be? It would strike me that you were heading down a path very much about work related to physiology of diving.
Yeah. And just to note, when I was looking at doing that research in the first place and looking at graduate programs, there wasn't a graduate program around for dive medicine or dive physiology. There were medical programs, so for medical students who are looking to do additional training, but there wasn't anything for PhD students. And so it took me a little bit of time to figure out where my proposed PhD work could potentially fit. So I had a proposal. We got funding for several years to do this research on biomarkers of decompression stress in divers. So I had Richard Moon, but Richard Moon was medical faculty, he was clinical faculty. And so he's like, I can work with you, but you're going to need somebody who has that PhD specific experience. And so I also had to find a department to fit under. So I started kind of feeling out different departments, and what I landed on at Duke was the Department of Pathology, which is essentially kind of the catch-all department for, it's like biomedical research, essentially. So anybody who was doing research that wasn't cellular per se, that didn't fit into some of the other major departments for graduate work. If they had a clinical bend to their research, tended to end up in pathology. So my colleagues did research on radiation exposure, different types of cancer. They did a little bit more clinically focused research. And so I went to Sal Pisa, who was the chair at the time, and he was like, "You have funding already?" Okay. I mean, he's like, "You're going to have to apply." I was like, "Yep, okay, we'll apply." And I joke that I would have never gotten in had I not had funding, but but I got into the program. And so that's great. But now I have to find a thesis advisor. I can have Richard as a co-advisor, but he can't be my primary because he's not in the department. And I was using a technique for my research called gene expression microarray, which was relatively new at the time. And Mike Datto was the pretty much only person or one of the few people in the department, even at the university, really doing heavy research around gene expression microarray. And he actually was teaching a biomedical research tools course. I can't remember the exact name of the course, but essentially it's like these are the different techniques and technology that you can use for the research that you all are doing. So it's kind of a mandatory course for our program. And so after the second or third lecture, where he talked about gene expression microarray, I went to him and I said, "Hey, I'd like to talk to you about the research I'm doing and see if you're taking grad students." And he's like, "Nope." He's like, "I'm not taking any grad students." And he just made the decision he wasn't going to take on any more grad students. He was just going to focus on clinical work and a little bit of teaching. And he was in a leadership position at the time, leading the clinical molecular labs at Duke. And so I was persistent and annoying again. I guess that's the theme throughout my history. But so I was annoying and I went back to him and I said, "Will you at least take a look at my proposal and give me feedback? Because I want to make sure I'm doing this correctly." Because gene expression microarray, the tricky piece is it's a lot of data. At the time, they're still trying to figure out, like, how do you responsibly handle that data with those large quantities, especially statistically. This was back in like 2008, so I was still relatively new. He's like, "Read these papers." So I read the papers. I came back. I was like, "Cool, I read the papers. Here's some questions I have." So we went back and forth for a little bit. And I was like, "Hey, would you reconsider taking me on as a grad student?" He's like, "Nope." He kept rejecting me. And so he even told the story during my thesis defense. So the whole department knew about it. Long story short, his wife, who was chair of cell physiology at UNC, she was like, "What are you doing?" She's like, "You've got a grad student that's got funding who wants to come work with you. She can help you, publish some things. You guys can do some cool work together. Like, you should take her up on this." So because of her, he took me on begrudgingly. And I say that we have a great working relationship. I still talk to him to this day. We published a lot of cool stuff together when we were working together. So we have the funding. I finally have a PhD thesis advisor. I have Richard Moon as my co-advisor. And then there's a continuing resolution. So the funding that was supposed to come in to support my diving related research got put on hold. There's a quote that says, "Brick walls are there for a reason. It makes you prove how badly you want something." I actually had that quote up on my desk as a grad student. So I went back to Mike and I said, "Hey, I still need to learn how to handle this type of data. Can we find another project that I can work on in the meantime until this continuing resolution clears with the government?" We started working on some cancer related data that he was looking at for a potential idea that he had, a hypothesis that he had, and then potential manuscript. And that ended up leading to my first manuscript as a grad student.
Yes, that's, again, a nice story of serendipity and, sort of one brick wall goes up and you can go around it or through it or over it.
STEM Talk is an educational service of the Florida Institute for Human and Machine Cognition, a not-for-profit research organization investigating a broad range of topics aimed at understanding and extending human cognition, locomotion, health span, resilience, and performance.
When you first arrived at IHMC, you had a couple of papers tied to this cancer research that you just talked about. One paper addressed how genes with bimodal expression patterns not only define clinically relevant molecular subtypes of ovarian carcinoma, but also provide ideal targets for translation into the clinical laboratory. And there was another paper also that looked at precise microarray-based predictions of tumor behavior in breast cancer patients. Would you talk a little bit about both of them, just a synopsis on each of them?
Yeah, so for time's sake, I'll just give the high level synopsis. But at the time, this new technique, this new tool called gene expression microarray, there are more and more groups that are looking at using it for developing clinical diagnostic markers or signatures. And essentially with gene expression microarray, you're taking a single sample and you get the expression level of thousands of genes in a single sample. So again, lots of data coming through. A lot of potential for variance that comes from places, from noise essentially too. And so the first paper that I talked about, that was the one where I really dug into gene expression microarray data, was focused on intratumor variance and breast cancer. So if you were to collect multiple samples from the same tumor, but from different areas of a tumor, do you see variance in what you're capturing in gene expression microarray? So either single genes that we know are targets for prognosis and diagnosis in breast cancer, so like estrogen receptor and progesterone receptor, and also HER2. And then also for not just single gene targets, but there have been some multi-gene signatures that have been developed for defining best treatment in individuals. And so did that variance that you saw across samples from different locations in the tumor, did it change or was it affected for those multi-gene signatures that were driving prognosis and also prediction of therapeutic? Really important to know, right? So you would hate to know that you captured a sample that's telling you information that's actually not going to help the whole tumor. And so this was published in Journal of Clinical Oncology. And long story short, we found that for really robust genes that are associated with prognosis in breast cancer and also for treatment decisions in breast cancer, like estrogen receptor, progesterone receptor, and HER2, those were stable across the entire tumor, regardless of where you collected that sample. Those multi-gene signatures tended to fall apart, because if you sample from a place with maybe more inflammation or sample from a place with just a different cellular makeup, it would really impact those multi-gene signatures. So that was really important for the field to know at the time. And then the bimodal expression paper, that was my second paper that was published for my thesis work. Same thing, we're looking at big data sets. These were all publicly available data sets from multiple institutions that had been capturing these data for the previous 5 years or so. And they had not just the gene expression microarray data, but then they also had clinical outcome, treatment, things along those lines. And so we were really just trying to maximize the data that was already out there. And just, again, for me, it was just learning how to really understand the data and handle the data. And so the bimodal expression study actually came somewhat from that first paper. When we looked across a population of patients and we looked at some of these single gene targets like estrogen receptor, progesterone receptor, we would see, if you plotted them across the population, you would see that they would have a bimodal distribution pattern. So essentially those genes were either quote unquote turned on or turned off in that sample. If you looked at most of the other genes would just have a normal distribution pattern. So it doesn't really tell you anything specific to prognosis or even treatment decisions. And so the question came up is, are there other bimodally expressed genes in different types of cancer, like estrogen receptor and progesterone receptor and HER2 in breast cancer, that could be clinically significant? And so we started to look across these big data sets and even learning how to merge those data sets. And, again, there's a lot of statistical work that goes into doing that. We were able to find a number of individual targets and also kind of multi-gene targets that were very distinct and showed a very distinct difference in both patient prognosis and also response to different types of treatment. We ended up publishing that paper as well as hopefully a starting point for others to be able to follow on and do further studies. So you don't have to do gene expression analysis, but essentially those targets could then be, proteins that you're measuring in patients when you collect samples from them. Really cool research to be a part of, really impactful, I think, for the community. And again, it just helped me understand better how to handle this gene expression microarray data.
That does sound like it would be useful and would lead to further research. Before you arrived at IHMC, you were collaborating with Dominic D'Agostino at the University of South Florida, who is also a visiting research scientist at IHMC. You are becoming known for your research on neuroprotection for people working in undersea and extreme environments, perhaps more generally. It was partially through Dom that you and I met, actually. Why don't you start by telling listeners what you remember about meeting me, and then I'll jump in and tell listeners what I remember about meeting you.
I was doing neuroprotection research at Duke University as a postdoc. And Dom and I knew each other from the diving medicine world. We had known each other for quite some time at that point. And he said, hey, there's this guy, Ken Ford, and he's interested in meeting some younger researchers who are kind of working in the field of extreme environment, performance, and health. And I gave him your name. So he might be reaching out to you. Long story short, you and I connected, Ken, and we met in DC. I was actually up there for an FDA meeting for a company that we were working with as part of my postdoc research. We were focusing on ApoE at the time, which we've talked about several times on this podcast. We were looking at a therapeutic target based on ApoE, and I just gathered that you were interested in the work we were doing in that space. And then you invited me down. Yeah, sure, I'll go to Pensacola. And then I was out on a boat with our friends Roger and Barbara Orth. That's the first time I met them. And you and Nancy came out with us. We spent the day not realizing how special that was, that everybody took the day to go out on the boat. And we saw the Blue Angels, and I got a window tour of downtown Pensacola and really got to meet a lot of the researchers at IHMC. I was able to eventually join you at a Blue Sky meeting, still not fully comprehending. Like, I think at that point we had talked about me potentially coming to IHMC, but I guess I was slow on the uptake. But I ended up accepting because I was like, this is just such a special place, and the type of research that we can do is so impactful. And so it was a pretty easy decision for me to make. And I will say this too, Ken and I found out that we have some shared contacts in completely non-research related worlds, like Paco. And we figured that out over dinner, which was a pretty funny coincidence.
Yeah.
But yeah, so anyway, so those are my memories about meeting you and then eventually joining IHMC.
Yeah, that largely coheres with my own memory. I remember on the boat ride, somehow, nobody knows how, one of the Blue Angels made a low pass right over the boat.
Yeah.
And that was good. And there was another young person on the boat who also had applied for work. And your reaction to it and her reaction were absolutely the opposite.
I think I said that's the sound of freedom.
Yeah. She said somebody should report them. Yeah. I said, okay, no need for further interview.
So there you go.
Yeah, I do remember.
Yeah, that's fantastic.
I do remember all of that. And you and Dom actually published a pretty significant paper that investigated how mitochondrial health, and I'm really fascinated by mitochondria, is a potential method for preventing oxygen toxicity during exposure to hyperbaric environments. Could you talk a little bit about this paper and some of the other work you and Dawn conducted during your time at IHMC?
Yeah, so that paper was actually a review paper, and I was also included Angela Poff, who's the lead author. And essentially, we were looking at the cellular consequences, the pros and cons, essentially, of hyperbaric oxygen exposure, with a special focus on mitochondrial health. And we were exploring how these processes mediate that balance of cellular protection versus damage. So it was a review paper, so it was really capturing a lot of the research that had been done to date. But again, I think it was a really impactful paper, and it was a fun one to work on with them. And then Dom and I ended up continuing, as I noted, we had known each other for quite some time. And so we ended up delving a little bit more into ketone studies for extreme environments, not just diving. So he'd been focusing on oxygen toxicity, but we also looked at impact of ketones in the cold water diving environment, and then also ketones for hypoxia. So looking at both cognitive and physical performance. And so it was a fun way to be able to merge the two areas of expertise, but also work with somebody that just have so much respect for and have been able to know for quite some time.
Yes, that was a very fruitful collaboration. While we were at IHMC, you also became an aquanaut. Shortly after you arrived, you were, as you mentioned once, helping me with the Blue Sky meeting that was focused on human performance, both in the undersea environment and the space environment. And we were looking at the kinds of lessons that we could learn from these two areas and how there might be crosstalk and how there might be application for future space exploration. And it was at that meeting that you met Bill Todd, who was the founder of the NEMO project for NASA. And NEMO, as I mentioned earlier, is a project that sends astronauts as well as engineers and scientists to live in an underwater lab that is the world's only undersea research center. Can you tell the story of how you became a NEMO crew member? And we'll talk more about NEMO in a few minutes.
Yeah, so going back to that microarray research that I was doing as both a graduate student and as a postdoc, I had some ideas around doing microarray based studies and some other what we call omic studies during a NEMO mission. Because I think one of the things with that Blue Sky is we're addressing kind of what research had been done from sea to space, and then where were the future areas of research of interest for Navy and NASA. So I talked to Bill Todd about would there be interest in allowing a researcher to come on site to collect blood samples before, during, after a mission for some of these omic studies, including microarray gene expression. He said, yeah, absolutely. Let's talk about it. In the meantime, I was putting together proposals to both NASA and the Navy to get that research funded. So it was December of 2015, a few months after we started talking about this research, that Bill called me up. He's like, hey, I just want to check in with you to see, what's the state of research, where are things with your studies for the project that's going to be kicking off next summer, the NEMO 21 mission. So I told him about the proposals and where things stood. And, we talked around the research piece of things. He's like, well, I've got another question to ask you. And Tim Broderick was actually on the call as well. So Tim Broderick is with IHMC now, and he had been a previous NEMO aquanaut. So he had actually been a crew member twice with NEMO and actually spent the most time underwater as a NEMO aquanaut. And so Bill comes back and said, so what do you think about for diving? And I said, I'll throw tanks on. I'll clean the bottom of the boat. I'll carry tanks. I'll do whatever you want me to do. And he said, so what do you think about becoming crew and living undersea? I still remember where I was. Like, I remember just sitting there, kind of my jaw just dropping, like the cartoon character. And he and Tim are trying to convince me how amazing it will be. And I was like, okay, just hold off. Like, you don't have to convince me. I'm in. Just let me know where to sign. So anyways, that was a very cool invite and definitely a life-changing experience for me to be able to join the NEMO mission in 2016.
Yes, those are great experiences. And for listeners who want to know more about NEMO and the Aquarius Undersea Laboratory, check out episode 19, where Tom Jones, a veteran NASA astronaut who flew 4 space shuttle missions, and he's also a senior research scientist at IHMC, interviewed Dawn about her experience as a crew member aboard NEMO. And as I mentioned earlier, Dawn was living in the undersea habitat when Tom actually interviewed her, which was cool. Little did you know at the time that a decade later, you'd become director of scientific research for DEEP, a United Kingdom startup with a mission to build modern subsea human habitats. And we'll talk a lot more about DEEP in a few moments. But as you look back at your experience on NEMO, are there any key takeaways from the mission that you think prepared you for the work that you're doing now?
I think the 2 biggest things, there's tons of takeaways. I could spend multiple podcasts talking about that, but I think the 2 biggest ones, one is just the impact of living subsea. As noted, I spent over 2 decades diving at that point of my life and have spent a lot of time underwater. Living underwater and being there 24/7 and truly being immersed and locked in with that environment for a sustained period of time. So my mission was 2 back-to-back 8-day missions. It was just incredible what you could see that I don't think you would traditionally see if you just drop in doing a single scuba dive, right? So I think it's been likened to if you're trying to explore the rainforest and truly understand the rainforest, if you rope in from a helicopter for 30 minutes and come back out, you're only going to get little bits and pieces of what's actually happening in that rainforest. But really being integrated into that environment for that period of time is just, it was incredible. And just, it really allowed us to kind of lock in with what's happening in that environment 24/7. And I've heard the same thing from other very experienced divers. I was recently visiting one of the Navy labs, and there are some folks there who have just storied careers and underwater work, diving, Navy diving, have dove out of every vessel you can possibly think of, spent a ton of time undersea. But they've also dove in an undersea habitat, and they said the same thing. Being able to look out the window, being able to go out and do these excursions of 6 or 7 hours at a time, it was life-changing for them, comparatively speaking. They're like, and I've spent tons of time underwater, but just having that ability to really integrate with the environment was such a change and made such an impact on them. And then the other big point, I think, that I learned in that mission is just how much work you can accomplish when you're diving in saturation. And so I can talk about that in a second, but essentially with this type of diving, you're equilibrated to the surrounding environment, to the surrounding pressure. And so when you go out to do dives, you don't have restrictions on your time, relatively speaking. So you can go out and do 6-7 hour dives and just work outside of the habitat. And so just really amplifies the amount that you can accomplish when you're doing those excursions and doing those dives.
Yes, living undersea like you did during the NEMO mission is certainly a different experience than, for example, living in a submarine. And you just mentioned saturation, and when you're in a habitat, you're in saturation. Can you talk a little bit about what happens to a person's body when they're in saturation? You talked about the implications of being able to work more effectively in that environment, but there are physiological issues as well.
Yeah. I mean, so there's a long history of studying saturation diving and how it impacts the body. Actually goes all the way back to the late 50s. There was research that was done that actually ended up feeding into some of the first undersea habitat programs. With that knowledge, we know that saturation diving, you have a point where the blood and tissues become saturated with the inert gas that you're breathing. So in our case, we were breathing air, which is 21% oxygen, 79% nitrogen. So essentially, that inert gas is the nitrogen, and our bodies essentially become saturated and become equilibrated to the equivalent pressure of nitrogen at that depth. So once you've reached saturation, then you're not considering the time restrictions that you would have with traditional scuba diving. So typically, if you went down to 50 or 60 feet, you would be able to stay down for, an hour or so before you have to come back up to the surface. That allows you to come straight to the surface with a little short stop on the way up. It really minimizes your risk of developing decompression sickness or the bends, and that's where bubbles form in your blood and tissues. And so you can do more technical dives where you extend that time on the bottom, like I was talking about the dives that the WKPP did back in the day. But again, it's going to start to lengthen that time to decompress. You're going to have to start breathing more complex gases. It just becomes riskier, and you still have relative time restrictions and what you can do. When you're in saturation, if I'm down there for 8 days, every day I could go out and do a 6 to 7 hour dive and come back, and I'm not going through decompression. You just go through that single decompression at the end of that saturation exposure. And that actually happens in a habitat. It happens overnight while you're sleeping. So they actually have somebody locked into the habitat with you, and they slowly but surely decompress that habitat. You're still sitting on the bottom, but they close it off so that they can reduce that pressure. And essentially, you're at the equivalent of what would be a submarine at that point when you wake up and you're at 1 atmosphere. So that's equivalent to the pressure that we're living at here on the surface. And so that's the big difference between submarines and habitats, right? So habitats, you're living at pressure versus the submarines, you're going to be living at 1 atmosphere. It allows for just increased efficiency on the bottom and also just increased safety. So, and there's a long history of using this type of diving in commercial diving and military diving. And so saturation diving isn't necessarily something new, but it's something that we want to continue to build on and better understand, especially since a lot of that research that was done, as I noted, was late 50s all the way through the 80s. We have a lot more advanced technology and more things that we can measure to really truly understand how saturation diving is impacting the body.
Absolutely. And let's talk about how you became the director of scientific research for DEEP. You were living in North Carolina, close to your family, and working as vice chair for research in the Department of Neurosurgery at the University of North Carolina. And you're also working here as a visiting research scientist. You pretty much had a great situation. Then all of a sudden, you get this call from someone who wants to introduce you to some people over in Britain. Want to discuss what happened next?
Yeah, I had a couple of friends who had been working for this stealth company. So I knew something was up in the UK with respect to diving, because I had a few questions that were thrown my way. They're like, I can't tell you anything, but we've got this question for you. They had actually been trying to connect me with these people from DEEP. So I knew that the company is called DEEP. It took about 6 months before we were finally able to get together. And I was actually in San Diego for a STEM outreach project that I was working on for the Navy. And my contact, Kurt Kroc, who's a world-class free diver, and he leads the human performance program for DEEP, he said, I want you to meet some leadership from DEEP. They're actually out there for Comic-Con, funny enough. So Kirk was on a panel for Comic-Con because he worked on the Avatar films as training all the free divers on cast and crew. And so this was kind of a strange blend of things. Like I was there for a STEM project and they were there for Comic-Con. But anyway, so we ended up getting together, found a location to sit down, and they put up on the screen the first slide, which shows the undersea habitat that they're building. And I just started asking questions. Again, curious Dawn started just asking questions left and right. So they're like, yeah, that's why we have you here. And so we went through the whole presentation. They didn't outright say, hey, do you want to join us? Essentially, I talked to Kirk after. I was like, this is amazing. I'm super happy for you. Let me know how I can help. And then he said, we're interested in bringing you on board. It took a few months of going back and forth. I went over to visit them and just figuring out, what that position would look like. And yeah, so director of scientific research for DEEP. And it was a hard decision to make. I was leaving a pretty comfortable academic position. I had, some good funding opportunities that we had locked into. We were actually studying the glymphatic system in extreme environments, but this just was something that I couldn't turn down. I was like, my question to myself is, would I regret not taking this? I was like, if I saw somebody else in that role a year from now, I would kick myself for not taking it. So I took the position.
Probably a good call on your part. Life is an adventure, and closing off some potential great experiences is usually a mistake.
Yeah. Same thing. So like when I joined IHMC, I had someone else offer me a position at Duke. And IHMC, there's just a lot of opportunities here. And so we always ask our guests on STEM Talk if they have any advice. And I think that's probably one of the other ones. I talked about following your heart, but even in these decisions, right? Following your heart and what feels like the right thing. Sometimes it may not be the most popular decision with everybody in your life. I can tell you when I took this position, everybody's like, you left a vice chair for research position at UNC, I would kick myself if I didn't. So I think that's really important to stick to your guns.
For sure, you made the right choice in my view. Deep is a company that's pioneering a new era of ocean exploration. Oceans, as everyone knows, covers around 70% of the Earth's surface and obviously hosts more life than anywhere else. However, in some regards, we really don't know that much about many aspects of the ocean. Can you give listeners a brief primer on why it's important to better understand our oceans?
Yeah, I'll just keep this short and sweet and to the point because I think we have some other podcasts around this too. But again, vital for life on Earth and provides essential resources. The oceans regulate the climate, support incredibly diverse ecosystems. They produce over half of the world's oxygen. They absorb very high amounts of carbon dioxide, and they have influence on weather patterns. So, I mean, you see all of that in our day to day, but I don't think people fully appreciate the role that oceans play in our day to day life, even if you don't live by the ocean, right? It impacts all of us.
Right.
And then beyond that, there's the impact on humans, looking at human economy through food production, transportation, recreation, and also offering the potential for renewable energy. So again, our oceans just are critical for our day to day life.
Deep has a mission to expand human subsea habitation through both research and new innovation. Can you tell us about some of the challenges associated with building these underwater habitats for humans? It has to have some technical as well as other sorts of challenges.
Yeah, so the technical challenges is really around building a safe pressure vessel. That's the most important part. And then the life support systems that are integrated with those habitats. But there's this long history of subsea habitats that actually started in the 1960s. And as I noted, the saturation diving research, so the focus on the humans living in those habitats, started in the late 50s and really carried on through at least the 80s, if not even since then. There have been some studies on saturation diving as well. Again, I think that we know these challenges, but also noting that there's a long history and we're really building on the shoulders of these giants that came before us. Right now, our big thing is taking those habitats to the next level, right? So we kind of got the basics down and understanding what are the important pieces to keep those habitats safe and the life support systems in those habitats efficient and optimized for the humans living in there. And so given that the last human-occupied subsea habitat was built in the 1980s, our big challenge right now is how do we evolve that? How do we take that to the next level, given that was 45 years ago?
Absolutely. This is probably before your time, but I remember in the 60s, the French oceanographer Jacques Cousteau, and he had this dream about building underwater villages. Of course, that didn't actually happen, but Cousteau was very motivating. His show, I think, attracted a lot of people to related subjects. As we were just discussing, Deep aims to create a relatively permanent deep-sea human habitat in the ocean. And you could think of it as kind of analogous to the International Space Station. Since 2000, the space station has allowed astronauts and researchers to live and work in outer space. And Deep wants to do a sort of similar kind of function, but in the ocean. And actually, there was an interview with you in Oceanographic magazine where you said you saw Deep as sort of an undersea version of that. When you think of the underwater habitat that Deep is building, it's called the Sentinel system. And the idea here is to allow people to live and work at a depth of 200 meters, which is 656 feet, for up to a month. Could you give listeners a sense of the new habitat that's under development known as Sentinel? What will this habitat be like?
Yeah, and I'm just going to touch on two other points that you made back there. That Cousteau dream, that's a big piece that's fueling what we're doing at Deep. So actually our founder said, hey, Cousteau was going to do this back in the 60s. How have we not done this yet? Especially since we've had a sustained human presence in space, since 2000. And that comment about NASA, I think one of the things to really drive home is that we're not just an engineering company that is building subsea habitats. It's not just the engineering piece, but we're also developing the training and the testing and evaluation capabilities and also doing the research that's required to make these systems safe and then also to keep the humans that are living in these systems safe and working to the best of their ability. We don't think we're NASA. We're not going to be NASA. NASA is its own entity. But I think just thinking of NASA in the sense that it's not just an engineering agency, that they have a lot of different facets to what they do to make a more comprehensive system in space exploration and science. And so we're planning to do very similar for subsea in that sense. For Sentinel, and there's actually a couple of different types of habitats that we're building, but Sentinel is essentially our flagship habitat that we're building that would have a depth capability of up to 200 meters, which is deeper than the previous habitats that have existed. Really excited about it because it's a much larger habitat, going to have individual staterooms or bunk rooms essentially. So instead of sleeping 3 layers deep in a bunk space on either side of the habitat where you're sleeping essentially next to everybody. And I've noted in previous conversations that when you're at depth and under pressure, people tend to snore. If they don't snore already, they snore louder, I think, than they typically do on land. And so being able to have your own space for good quality sleep, I think is going to be really important, especially if we're thinking about optimizing performance for longer duration missions. And the other thing I'm really excited about is that given this larger habitat, which is going to be half the size of a Boeing 777. So if you think about like top to bottom width and just the size of that, comparatively speaking to some of the previous habitats, is pretty substantial. And so we're going to have internal laboratory capabilities as well. And I'm really excited about that because thinking about what kind of samples and what kind of research we can be doing at depth. So not just collecting things and then bringing them to the surface, and then having that time delay, and then also the decompression of those samples, which impacts those samples. So whether that's marine samples or human samples or whatever that looks like, I think it's going to be really exciting to have that capability from a scientific perspective.
Absolutely. And as we discussed earlier, the Sentinel system will enable researchers to work at a depth of up to 200 meters. And as you said, that's actually quite deep. And this represents the epipelagic zone, also called the sunlight zone, naturally enough. And it's the deepest point at which light penetrates the ocean and where a vast majority of marine life is believed to exist. To put this in perspective, the average depth of the ocean is 3,688 meters. We really don't know very much about what exists below the 200-meter level, do we?
No. And honestly, we don't really know that much about the zone between 50 and 200 meters. Really, you could say that about the entire ocean, but in particular, anything deeper than 50 meters. Typically, what we know about anything deeper than 200 meters is going to be from things like ROVs and AUVs and submersibles. So there's a huge gap in knowledge. And honestly, for the habitats that we're building, there's a big target of that 50 to 200 meter range. So essentially, up to 50 meters, you can get a fair bit of research done using traditional scuba diving, even maybe a little bit deeper technical diving. And then deeper 200 meters, you tend to switch to things like ROVs, AUVs, submersibles, which have their own restrictions. And especially for something the size of the ocean, you're really just kind of capturing small bits and pieces of what's out there. And so being able to focus on that 50 to 200 meter range is really our sweet spot for our habitats and will allow for us to hopefully over time get a better understanding of what exists in that range.
So NASA has a long history of using the sea as a loose analog for some aspects of space. And as you well know, there are a lot of interest right now in NASA's ambitions for human exploration, the moon, and maybe potentially someday Mars. But most people have a very limited appreciation of how difficult this actually is. Will the Sentinel and subsea habitats help in the preparation for deep space missions such as Mars or even a return to the moon?
Yeah, it's another great question. So there's actually a technical report within NASA that was developed looking at different types of analogs. So for training and research, looking at ground-based analogs or Earth-based analogs for being able to do research. And this is specific to deep space exploration. The best analog out there was considered the International Space Station. So no surprise on that one. But then the next one out of all the different analogs that they have, isolation analogs, other types of environments where they do this kind of testing, It was the Nemo mission. It was that undersea mission where they saw the highest fidelity with respect to being able to simulate a high stress environment. So when you talk to even astronauts that are out there who have been in space and have also done Nemo missions, they'll tell you that having that psychological stress of knowing that you are in this high risk environment, that the decisions that you make are not just impacting you in that moment, but also the health of your crew and you is very much like space. So just noting that you can't just come up from a saturation dive if anything goes wrong. You have to stay down or then you risk very serious decompression sickness or death if you come straight to the surface from a saturation dive. So they always have that in the back of their mind. And then there's the psychological piece of being isolated, relatively speaking, and working in a small environment with a small crew. So there's a reason that they've been using Nemo missions from 2000 to 2019 as a pretty comprehensive analog. And I think we've had a lot of discussions, not just with NASA, but also with other commercial space companies who have interest in using a subsea habitat for different types of training and also research before their crews go into space. And so I think that's going to continue just in that sense, using this as this relatively high stress environment for crews to train in and also for that research to be conducted before you're looking at longer duration missions.
So we've been talking about Sentinel and habitats and very exciting. What other areas of research are you excited about diving into, so to speak? Sorry about that. Once your first habitat is actually in the water.
We've got a lot of different research ideas. Our near-term research that we're building up right now, one, we're building out a longitudinal health database for our crew. So that's something that we're really excited about because it's not just going to be like historically where you had the saturation divers from military and commercial industries, but now we have a broader population. Kind of, if you think about like commercial space, the same thing. You have a bit of a broader population going in to do these dives. And so we're going to be capturing data from them before, during, and after the mission. That includes imaging and that also includes biobanking samples for future analysis. We're going to be involved in some early coral, sponge, and other species restoration efforts and working with different partners at some of our different sites, and that which we're really excited about. Human-machine teaming. So I know there's a lot of interest in potentially working with researchers at IHMC in this space, but that's something that keeps coming up in our discussions of how do we keep the human in the loop as we're thinking about undersea exploration and research. And so I think that's going to be a critically important area for us to delve into, especially as we have humans that are living in that undersea environment. And then marine pharma is another really exciting area. So thinking about using the saturation dives and being able to use that extended time on the bottom to really explore different areas, capture peptides, and really get a feel for what's out there instead of trying to do that on these single exposure scuba dives. So those are probably the biggest areas of research that we're most excited about, I would say, in the first year or two of us getting in the water.
Very interesting and very promising, I might add. DEEP has a testing and training facility in the United Kingdom close to the border between England and Wales. Could you give us a brief overview of that campus and its purpose?
Yeah, so that facility actually was a quarry that was used for recreational diving for quite some time and also other types of dive training. Our company bought it out, and it's kind of funny because it was bought out and closed down, and there are all these rumors around Chepstow, which the campus is located close to Chepstow. You know, the community is like, oh, who are they training? What kind of research is going on over there? What's the work doing? Some of the stories are pretty funny, actually. But essentially, it's a confined water facility. It has up to 80 meters of depth. So right now we have submersibles. Our 2 submersibles are over there right now. We have other technology that we're putting into the quarry for testing and evaluation. For some of our partners, it's a great place to go. So unless you have lightning, you can go use that facility for any kind of testing, diving, training that you need. And so it's really nice to have that kind of dependable facility for that type of work.
Sure.
And then as we're building out our habitats, it's very close to our first manufacturing facility, which is in Bristol, United Kingdom, and it allows us to do some in-water testing as well.
I can see the value of that. In addition to the collaborations we mentioned earlier with IHMC, we hear that you all have recently established a collaboration with the Florida Institute of Oceanography. And it seems like you might be building more of a presence these days in the United States and in Florida in particular.
In the U.S., we've set up a separate U.S. entity, and that was as of late 2024. And we are planning to put our first habitat down off the coast of Florida in Q4 of calendar year 2025, which is really exciting. And there's more information to come on that. But yeah, so there's a long history of subsea habitats off the coast of Florida. And so this is a great place for us to get started with everything. That collaboration with FIO essentially allows us to work across all the different state academic marine programs and the different marine institutes, because they're all part of that consortium with FIO.
Projects like Sentinel are expensive and will require continuous funding for sustained operations and also for research continuity. Can you tell us a bit about DEEP's funding model?
Yeah. So we were founded by a single individual who made some money earlier in life and said, hey, why are we not living subsea when we've had sustained human presence in space since 2000? And Cousteau was planning on this back in the 60s and it still hasn't happened. He has a huge passion for the ocean. He also understands the importance of accessibility and getting eyes beneath the surface of the ocean. So not just on screens, but actually giving people that exposure. And so he really wants to increase that accessibility for humans undersea to better understand that environment that we're asking them to not just understand, but protect over time. He's essentially set this up as a generational funding model. So the thought is that we're not going to do all the work we need to do for DEEP in our lifetime, that this is going to be multiple generations to really have that sustained presence subsea and to have more accessibility subsea for researchers, explorers, educators, anybody that wants to get undersea and get their eyes beneath the surface of the ocean. And so it's been really fun to work with somebody who has that kind of vision. And then, we're building this out as a commercial entity, but the idea is that with that commercial piece, we're always feeding back into the growth of DEEP over time.
It's an interesting approach. As if you don't have enough to do, I understand that you're the one that'll be in charge of establishing DEEP's first U.S. office in Raleigh, North Carolina. So that's just yet another thing on your plate.
Yeah, that would be my house. So that one's pretty easy. Probably the hardest thing with that one is keeping track of my cats and just, keeping things balanced in the house, on the home front. But no, it's great. Really, honestly, more than anything, I'm on the road a lot right now, just trying to get the word out about what we're doing.
I can imagine. Talking
to different researchers, talking to different funding agencies, making them aware of who we are, but also thinking about what potential collaborations look like down the road.
Yes, and I'm sure that you're good at sharing the passion and the vision that DEEP has. And you must be very busy, but it's also exciting. So that makes it all possible. If it wasn't exciting, it would be exhausting, I'm sure.
Yeah. And you can probably hear it, from me. I love what I do. It's because I saw and I had that personal impact of being in that undersea mission during Nemo. I understand what that could do for other people, and I think the impact that it could make in the research community. So it's a very easy message to share, and I can tell you, being on the road, it's just continuous excitement from the communities that we're talking to about what we're doing. So that's really special.
It is.
STEM Talk. STEM Talk.
STEM Talk. STEM Talk.
So Ken, what did you think? How did I do?
It was terrible. No, you did great. I knew you would. DEEP is such an exciting thing. I think everyone should keep their eye on it. There's really no project like it in the world. And so I do think it's fascinating.
Oh, thank you so much, Ken. Yeah, I feel very honored to be a part of all of it and still get to work with IHMC as a collaborator. So that's fantastic.
It is, and we're happy about that. And, when one thinks of DEEP, it's really both an adventure and a discovery platform, expanding the scope of human activity and habitation. And again, the analogy to space is right there, right? It's very, very strong analogy. Yet this is right here on Earth, where we, in some ways, know less about it than we do about the moon, for example.
Yeah, that's so true, Ken. Yeah, and so again, thank you so much for giving me the chance to talk a bit about what we're doing.
It was wonderful, and thank you for doing it.
So this is Dawn Kernagis signing off for now.
And this is Ken Ford saying goodbye until we meet again on STEM Talk.
Thank you for listening to STEM Talk. We want this podcast to be discovered by others, so please take a minute to go to iTunes to rate the podcast and perhaps even write a review. More information about this and other episodes can be found at our website, stemtalk.us. There, you can also find more information about the guests we interview.